Right. Hello again, everyone. Right. This is the last lecture in this series of lectures in biochemistry, and we're going to finish off with looking at the different stages of cellular respiration. Now we looked at cellular respiration in general in the last lecture, so we touched upon a few things that we're going to talk about in a bit more detail in this lecture. Okay. So as is customary, let's look at the learning outcomes. Okay. So in this session here, we're going to talk about all the different stages of cellular respiration. Okay, and one of the stages of cellular respiration is glycolysis, and we're going to talk about this in the most detail. Okay, there are other stages which we'll just sort of gloss over and maybe just mention a few things, but it's really glycolysis we're going to talk about in much more detail. Okay. Um, and we're also going to look at the main purpose of cellular respiration is to produce ATP, and different stages yield different amounts of ATP. Okay. We're also going to look at what stage carbon dioxide is released as well, is produced and released. Okay. And also look at regulation as well. So there's all these different stages of cellular respiration, and each one of them is subject to regulation. Okay, so we look at what steps are taken to regulate cellular respiration. Of course, sometimes we need to produce lots of ATP in the case of exercising or any kind of physical activity, but then there are other times when we are resting, so we don't need to produce as much ATP. Okay, so those stages have to be regulated to accommodate these changes. Okay, and also again look at redox reactions in ATP generation. Before we actually go into and talk about cellular respiration in more detail, let's just have a quick preview of the different stages of cellular respiration. Okay, so first of all, we start off with glycolysis. Okay, so this is the first stage, and this happens. It doesn't happen in the mitochondria; it happens in the cytoplasm. And the main purpose of this is really to break down the glucose into two molecules of pyruvate. Okay, so if you remember, we're oxidizing glucose, so we're breaking it down. Okay, by oxidizing it. But the initial breakdown products after glycolysis uh, results in the production of pyruvate or pyruvate. Okay, and pyruvate is a 3 carbon molecule, and glucose is a 6 carbon molecule. So we're splitting it. We're splitting glucose and modifying it to produce two molecules of 3 carbon pyruvate molecules. Okay, pyruvate then is passed on to the citric acid cycle. There is a transition stage here as well, which I'll come on to at a later point, and that's the production of acetyl CoA. Okay, but so pyruvate gets converted to acetyl CoA in the transition stage. Okay, but that's a relatively small step. Okay, a very important step, but it's a small step compared to glycolysis in the citric acid cycle. Okay, so acetyl CoA gets converted from pyruvate, and then it enters the citric acid cycle. Okay, or the TCS cycle, the Krebs cycle, they're all the same thing. Okay, and this basically completes the breakdown of glucose. Okay, so it completes the breakdown of glucose. One of the main reasons for the citric acid cycle is to produce very important molecules. Okay, which are then used in the oxidative phosphorylation pathway, the final bit, and it's these important molecules that are produced not only in citric acid cycle but also in the glycolytic cycle, as well as the pyruvate dehydrogenase stage as well, the transition stage. Okay, so it's molecules, very important molecules produced. It's not. I'm not referring to ATP here; they're important, but it's other important molecules such as NADH and FADH2. These molecules are produced in the citric acid cycle mainly. Okay, and these molecules are then used to generate ATP. The vast amount of ATP that is produced during the oxidative phosphorylation pathway. Okay, so So the first stage, glycolysis, and like I said, it it's basically breaking the 6-carbon molecule of sugar. So it's 1, 2, 3, 4, 5, 6, right? Or we actually start from here. 1, 2, 3, 4, 5, 6. Okay. So we basically modify that and then eventually split it up into two. Okay. And then modified it further to yield 2 molecules of pyruvate. Okay, so 2 times 3 is 6, which counts for the 6 carbons here. Okay, so this is catalyzed by 10 different steps. Okay, meaning there are 10 different enzymes involved here. Okay, it occurs in the cytoplasm, and this is the initiation stage, if you like, of cellular respiration. Okay, um, and it does, and this this stage here doesn't require, okay, or this pathway or this cycle rather, doesn't require oxygen. Okay, so it can happen. So when we talk about anaerobic respiration, okay, this is the first stage of anaerobic respiration as well. Okay, and then once we get the pyruvate is then modified or converted rather by to acetyl CoA. So there's a few steps that go on here, the transition stage here. Okay, and it gets converted to acetyl CoA, and then acetyl CoA enters the TCA cycle, the citric acid cycle, also known as the tricarboxylic acid cycle of the TCA cycle, or the Krebs cycle. Okay, so all these different names mean the same thing. Okay. Now, this is essentially an aerobic process, although you won't see any arrows carrying oxygen here. Okay, but because it's tightly linked to the third stage, the oxidative phosphorylation pathway. Okay, and that is um, that absolutely requires oxygen. Okay, and because it's linked to that, if the oxygen runs out for the oxidative phosphorylation pathway, this doesn't work as well. Okay, so it is an aerobic process, and it takes place at the mitochondria. Okay, so unlike glycolysis, which takes place in the cytoplasm, citric acid cycle and the oxidative phosphorylation pathway take place in the mitochondria. Okay, and basically its purpose is to oxidize completely the original glucose, which was converted to pyruvate in the glycolysis pathway, into a 2-carbon compound. Okay, oxaloacetate. Okay, and the main reason for this pathway is really to supply the third stage, the oxidative phosphorylation pathway, with the high energy electrons. Okay, and the way it does this, it does this by generating these molecules here: NADH, NADH, FADH. Okay, these are the high energy electron carrying molecules. Okay, and it's these that get passed on to the third pathway, which allows for the generation of ATP. Okay, this pathway, the citric acid cycle requires 8 different enzymes. Okay, so already we're up to more than 18 enzymes, because the transition pathway also has a couple of enzymes involved. Okay, and then we come to the final stage, the oxidative phosphorylation pathway, stage 3. Okay, so this is where those high energy electrons are carried by NADH, and it's these molecules, NADH and FADH2, which pass on these high energy electrons, and it's these high energy electrons that are used to pump hydrogen, okay, through these different protein complexes, complex 1, 2, 3, and 4. Okay, 2, 3, and 4. It's these complexes, especially 1, 3, and 4, that use these electrons, these high energy electrons, to pump hydrogen into the intermembrane space of the mitochondria. Okay, so and it's these hydrogens then, these increase in hydrogen ions on the intermembrane space that then come back through diffusion through the ATP synthase complex, okay, which then drive the energy that's that gets drawn from this diffusion. It drives the regeneration of ATP from ADP. Okay, and this is done by a a process called chemiosmosis. Okay, and it's basically the diffusion of this high concentration of hydrogen ions on the outside, driven to the inside by diffusion. Okay, so we've got a high concentration somewhere, low concentration on the other side. Things move through diffusion, and it's this kinetic energy that's provided by this movement of hydrogen ions that drives a rotor, a mechanical rotor in here, which provides the energy then to regenerate ADP to produce ATP. Okay, so those are the three main stages of cellular respiration. Okay, so just a bit of repetition here. So glycolysis, okay, is the splitting of sugar. So lysis means breaking up. Glyco refers to sugar, so it's the splitting of sugar. Okay, so glycolysis is the breaking up of sugar to break down molecules of pyruvate. Okay, now glycolysis occurs in the cytoplasm, so we've already mentioned that. Okay, but it has two major phases. There's what we call an energy investment phase, okay, and an energy payoff phase. Okay, so in other words, you have to put energy in. You have to put energy in to help destabilize. It's a bit like the activation energy to help to destabilize the glucose. Okay, glucose is a very stable molecule. To destabilize it, to be able to extract the high energy electrons in glucose, we have to destabilize it. Okay, so you have to put energy in. Okay, and so putting some energy in is called the energy investment phase, and then subsequent to that, you destabilize it more and more, and then it starts to pay off. Okay, then we start to generate ATP. So even in glycolysis, we can generate ATP. It's just that we invest 2 molecules of ATP and we get 4 out, so we get a net production of 2 ATP. Okay, and glycolysis occurs like I said, whether or not oxygen is present. Okay, so we don't need oxygen for this part. Okay, now this is the whole process of glycolysis. So you can see over here 10 different enzymes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, so 10 different enzymes. And you can see the products which are formed. Okay, you've got all these different products, and like I said before, we need to destabilize it gently. We need to destabilize glucose gently, because if we did it all at once, the amount of energy released would be explosive, and the heat generated would be too big for our bodies to cope with. So the breakdown of glucose is done gently through a stepwise process, hence the number of enzymes that are required to do this. Okay, so that's the whole process, and we're going to go through this now step by step. Okay, so we're going to focus on glycolysis. Okay, so the first step, okay, is the conversion of glucose to glucose 6-phosphate. Okay, now if you remember from the last lecture, I talked about hexokinase, the enzyme responsible for beginning the destabilization of glucose. And how do we do this? We take a phosphate group from ATP, okay, a high energy phosphate group, and attach it to glucose. Okay, now it's attached to the 6th carbon, so 1, 2, 3, 4, 5, 6. So it's attached here to produce glucose 6-phosphate. Okay, and although I've drawn it here as in a linear form, in actual fact it exists as a circular form. Okay, so it exists as a circular form, and this hexokinase enzyme requires magnesium, again, if you remember from the last lecture, we saw how magnesium is involved in the catalytic site of hexokinase to shield the excessive number of all these electronegative oxygens. Okay, so you need to shield these electronegative oxygens to be able to break one of the phosphates free from ATP. Okay, so this is part of the what we call the energy investment phase because we need to put energy in. Okay, so the phosphate group here from ATP, the energy here is then put into this part of the glycolysis pathway. Okay, so it requires energy. There's another stage here which requires energy, and so this bit here is the energy investment phase, but we'll come on to that in a second. Okay, so one of the phosphoryl groups or one of the phosphate groups is from ATP is transferred to glucose to produce glucose 6-phosphate. Okay, and it's done by this enzyme hexokinase. Now, incidentally, this product, glucose 6-phosphate, okay, can actually inhibit hexokinase. Okay, so the chances of that happening is if you get too much production of this. Okay, so too much production of this, okay, which then results in the production of all these subsequent molecules. But if this starts to build up, that means the pathway is going full speed ahead. So if this builds up, there's too much of it, and it's a signal that we're already producing too many of these molecules. So then, in that case, glucose 6-phosphate then has a bigger chance of feeding back negatively to hexokinase and inhibiting it. Okay. Another feature about the addition of a phosphate group, okay, is that it makes the glucose very charged because you've got this highly electronegative phosphate group. It's highly charged, okay, and if you remember, charged molecules cannot enter or leave the cell by themselves, not by diffusion. There's special channels for charged molecules to leave and enter the cell. Okay, so by adding a phosphate group, to the glucose molecule, you're making it highly electronegatively charged. Okay, and by doing that, you're trapping the glucose 6-phosphate inside the cell. Now, we have special channels on the membrane, okay, glucose channels that allow glucose to enter and also leave the cell. Okay, we want glucose to remain in the cell. So by adding a phosphate group, not only are you destabilizing the glucose molecule, but you're also preventing its loss from the cell. Okay. So the whole process requires magnesium. Okay, magnesium is required for hexokinase. It's a cofactor. Okay, hence the reason. So just relating this to diet, if you don't have enough magnesium in your diet, and magnesium usually comes from fruits and vegetables. Okay, so if you don't have enough magnesium, okay, it can have an impact on these enzymes which perform these really important processes. Okay, so hence the requirement of minerals in general, okay, in your diet. Okay, so another interesting observation is that hexokinase, there are different forms of hexokinase, 1, 2, 3, and 4. And these different forms of this enzyme, okay, are different, obviously, but also in terms of how much they're expressed, in which cell types they're expressed in, and how they're regulated. Okay, how they respond to regulation. So they're all quite different in those respects. Okay, now hexokinase. There's one form of hexokinase which doesn't respond too well to the inhibition of glucose 6-phosphate. Okay, And this form of hexokinase is you can find expressed in high concentrations in tumor cells. Okay. Now, one of the features of tumor cells is that, or one of the features of tumors in general, is that they're a bit disorganized. Okay, not bit, they're largely disorganized. So they don't have access to blood vessels like other cells do. Okay. Now, as you know, blood vessels bring in oxygen and other nutrients. Okay. So because they don't have lots of oxygen coming in. Okay, and remember, this whole cellular respiration pathway, except glycolysis, is an aerobic process, so it requires oxygen. So if it's not getting enough oxygen, we're not tumor cells aren't really producing lots of ATP through the citric acid cycle and the oxidative phosphorylation pathway. So they have to make do with this glycolysis pathway largely. Okay, and so hence the reason why they produce lots of hexokinase, which is not so inhibited by glucose 6-phosphate, so it continues to drive the reaction. Okay, so although we can put energy, we need to put energy in, as you will later find out through the investment, sorry, not the energy payoff phase, that we get more ATP produced than we need than we actually put in. Okay. Okay. One more thing about the hexokinase enzyme. An interesting thing about hexokinase is that because it requires ATP, okay, it requires ATP. It needs its best location would be where ATP is produced in large quantities. Okay, and so what you find is that hexokinase is actually physically linked to the mitochondria, to the outside of the mitochondria, because remember the mitochondria is producing ATP in the inside and then it's pumping the ATP out. Okay, and hexokinase just sits outside, linked to the mitochondria. Okay, so it has ready access to ATP. Okay, right then, and so the next stage is mediated by phosphoglucose isomerase. Okay, so this this molecule here, glucose 6-phosphate, is converted to fructose 6-phosphate. Okay. Now, what do we know about the difference between glucose and fructose? Glucose is an aldehyde, and fructose is a ketone. Okay. And what we can see over here, that glucose is an aldehyde, CHO group here. Okay. But now it's isomerized to convert the aldehyde into a ketone at carbon number 2. Okay, so we get a ketone. Okay. So we just go from a glucose form to a fructose form, and that is an example of an isomerization reaction. Okay, so phosphoglucose isomerase, that's the enzyme which mediates this isomerase reaction. Okay. Now, I said, didn't I, before that these all exist in cyclical forms. Okay, and they do, but in order to perform this isomerization reaction, we need to linearize the molecule first before we can actually introduce this ketone group here. Okay, but then once the ketone group has been introduced, it's been modified somewhat, and then it becomes recircularized. Okay. Okay, and then the third stage is again one of the stages which requires ATP. So it's another stage where energy needs to be invested. Okay. What you'll notice is the naming here. So the first one was a hexokinase. It's a kinase, and this is also a kinase. And if you remember, I did mention in the previous lecture what a kinase does. A kinase does. Okay, it's responsible for phosphorylating molecules. Okay, so it takes a phosphate group from somewhere, usually ATP, okay, and attaches it to another molecule. A phosphate group is a highly energetic molecule because of the all these oxygens being present in close proximity. Okay. So another energy investment phase, okay, mediated by this enzyme here, phosphofructokinase. Again, it's a kinase. It requires ATP, so it requires magnesium. So, and this another enzyme which requires magnesium. Okay. One of the interesting things about phosphofructokinase is that it helps speed up glycolysis. Okay. So not only do we get in tumor cells, do we get an increase in hexokinase production, but also an increase in phosphofructokinase production as well. So you get an increase in these two different enzymes in tumors. Okay. Now, what you'll see, what this enzyme does, of course, it's a kinase. Okay, it's phosphorylating the uh, uh, the molecule. So remember, before we already had a phosphate group already attached. So and what was the purpose of the phosphate group is to destabilize it. Okay, give it some energy to destabilize it. Now, if we add another molecule, this is what phosphofructokinase does. We've added another phosphate molecule. It's really destabilize the molecule even further. Okay, so now it's really all sprung up. Okay, and it's just wobbling because it's got so much energy in. Okay, so this molecule here is then becomes a fructose 1,6 biphosphate. Before it was a fructose 6-phosphate, whereas the phosphate group was only attached to the sixth carbon. Okay, carbon number 6. Now it's got two phosphate groups, so biphosphate, one on carbon number 1 and one on carbon number 6. So hence the name fructose 1,6 biphosphate. Okay. One of the interesting things about this enzyme here, okay, it's one of the hexokinase is a regulatory step, but also phosphofructokinase is another regulatory step. Okay, and ATP, if the cell is behaving normally, ATP can actually inhibit this enzyme. Okay, what's the purpose of this enzyme is to eventually lead to the production of ATP at some point. Okay, so if there's lots of ATP present, we don't need to drive this pathway. So ATP then, if there's lots of ATP knocking about, it will inhibit phosphofructokinase. Okay, so the more ATP you have, the bigger the chance that this enzyme will be inhibited. Okay, but then if you don't have lots of ATP floating around and you have lots of AMP, adenosine monophosphate, or even adenosine diphosphate, okay, these molecules, AMP especially, will increase the activity of this. Okay. Okay, and then we come to the fourth stage. Okay, and this is kind of the lytic phase. So remember, before we produced fructose 1,6 biphosphate, and what did we say? 2 phosphates. You're really putting so much energy in to really destabilize it. Okay, now it's ideal for breaking it up. Okay, and this is the lytic stage. Okay, this is where we get the production of 3 carbon 3 carbon molecules. Okay, so this is done by fructose biphosphate aldolase. Okay, and we get the production of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay, now so this fructose 1,6 biphosphate is cleaved to produce 2 different trioses. Okay, these are both trioses, 3 carbons. Okay, dihydroxyacetone phosphate, which is a ketone, okay, and a glyceraldehyde 3-phosphate, which is an aldehyde. Okay, ketone there and an aldehyde there. Okay, so this is what we call the lytic stage or the glycolytic stage. Okay, this is where we actually break the glucose down to produce 2 molecules of 3 carbon sugars. Okay. Now the thing about this molecule here is that it can't go any further into the dihydroxyacetone phosphate cannot go into the later on into the glycolysis pathway. Okay, the only molecule that can proceed further is this molecule here, glyceraldehyde 3-phosphate. Now, that's not really a problem because we can convert dihydroxyacetone phosphate to glyceraldehyde 3-phosphate by using this enzyme here, triose phosphate isomerase. Okay, so this enzyme here converts dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Okay, and that's compulsory in order for this molecule to be to enter to enter further the glycolytic pathway. Okay. Okay. So the first bit was the energy investment phase. Okay. The second half of the pathway is really the energy payoff phase. Okay. As you can see here, we are now generating ATP now. Okay. So before we needed to put 2 molecules of ATP in. So for every molecule of glucose, we had to put in 2 molecules of, we had to input 2 ATP molecules. And now for every glyceraldehyde 3-phosphate molecule, we're producing 2 molecules of ATP. But remember, we get 2 of those. Okay, so rather than producing 2, we're producing 4. So we put 2 in and we get 4 out. Hence, why we get a net production of 2 ATP molecules. Okay. Now, another benefit to this part of the pathway is that we start to generate NADH. Okay, NADH is one of those really important molecules that helps to carry high energy electrons. Okay, so we are actually reducing NAD, okay, to produce NADH. Okay, NADH was reducing its gain of electrons, so NAD gains an electron. Okay, and so as it gains an electron, it becomes a bit unstable, and then the hydrogen comes in to help to sort of stabilize it a little bit. Okay, so it's a way of carrying electrons, but in a very stable way. Okay, so so this is this is another one of the advantages of this pathway. So not only does it produce ATP and okay, but we also get the production of these very important electron carrying molecules. Okay. And of course, these are the NADH will be required for other pathways later on in cellular respiration. Okay, so this whole production of NADH is what we call it is part of a coupled reaction. Okay, now if you look at this, if you look at glyceraldehyde 3-phosphate, it's a phosphate group attached. Okay, now this enzyme here, glyceraldehyde 3-phosphate dehydrogenase, is responsible for reducing NAD to NADH, but it's also responsible for using the energy from here, okay, using this inorganic phosphate, okay, as well, to attach another phosphate group. Okay, so this is not a kinase. Okay, remember what a kinase does. A kinase is an enzyme which phosphorylates molecules. This is not exactly a kinase. Okay, but it does harvest some of the energy, okay, from this reduction reaction to actually phosphorylate glyceraldehyde 3-phosphate, okay, to form 1,3-biphosphoglycerine. Okay, so remember, we've got this phosphate group already attached as from before, okay, but now we've added another molecule of phosphate. Okay, so what do phosphates do? They destabilize molecules. Okay, so phosphate molecules destabilize molecules. So we had 1, so this molecule is already destabilized. By adding 2, we're again really destabilizing the molecule. Okay, and this is an important step because Okay, we want to extract as much energy as possible. Another important thing to remember is that in order to drive this process, we need NAD. Okay, now we only have a limited amount of NAD, just like we have a limited amount of ATP in our cells. Okay, we only have a limited amount of NAD. So uh, we get the production of of NADH rather. Okay, this NADH then is used in the oxidative phosphorylation pathway. Okay, and to regenerate NAD. Okay, and so if we don't get enough NAD being produced from the other subsequent pathways, glycolysis can be stopped. Okay, so we need NAD to be regenerated in order for glycolysis to keep on going. Okay. Now the interesting thing is that NADH gets oxidized to produce to generate NAD, but only if energy is being produced, or if only if ATP is being produced. Okay. If ATP isn't being produced, NAD isn't being generated. And if ATP isn't being produced, it's because ATP is probably not needed. Okay. So it makes sense that NAD uh, wouldn't be available because there's already enough ATP being produced. So glycolysis wouldn't be necessary then. Okay. So coming back to this, we've got this molecule here with two phosphate molecules attached to this, really destabilizing this, and this is just basically dying to give off some of the energy. Okay, and so this is what happens then. Okay, um, one of the phosphate groups. Okay, one of the phosphate groups is then used to phosphorylate ADP, adenosine diphosphate, to produce adenosine triphosphate. So there we are, we get the production of ATP. Okay, and it's this enzyme here, phosphoglycerate kinase. Phosphoglycerate kinase, again, it's a kinase, okay, uh, which phosphorylates ADP to produce ATP. Okay, so this is the first ATP molecule being produced. Okay, and what we have as a product of that reaction is 3-phosphoglycerate. Okay, 3-phosphoglycerate. Okay. Right then. And then the next reaction, okay, it's basically it's to help again to destabilize the product even further. Okay. And the way this works is by kind of mutating this molecule by mutating 3-phosphoglycerate. And what we're doing is when we say mutating it, what we're doing is we're shifting this phosphate group from this third carbon onto the second carbon. Okay, so now we're bringing this highly electronegative component closer to this carboxylic acid component. Lots of oxygens here, lots of oxygens there. You're bringing them into closer proximity, which really makes the whole molecule really wobble. Okay, so it's dying to give off the energy. Okay, now what we can do to destabilize it even more, okay, is remove is perform a reaction through this enzyme here, enolase, okay, which removes a molecule of water from 2-phosphoglycerin. Okay, and it produces this molecule here, phosphoenolpyruvate. Okay, now when we remove a water molecule, we go from 2-phosphoglycerate to phosphoenolpyruvate, and this molecule here is extremely unstable. Okay, it's just how the atoms are arranged and how the electrons are arranged between themselves that makes this molecule really unstable. Okay, and now this molecule is again dying to give off the energy, okay, in the form of this phosphate group. Now, one of the interesting things about this phosphoenolpyruvate, okay, is that it has a much bigger potential for transferring the phosphoryl group. Okay, so this ATP, ATP has an energy in which it gives off a phosphoryl group. So it has a phosphoryl group, a phosphoryl transfer energy. Okay, ATP has, and ATP is a high energy molecule because the phosphoryl transfer energy is quite high. But PEP, phosphoenol pyruvate, has an even bigger phosphoryl transfer energy. Okay, so it's this will give off the phosphate group even much more easier than ATP does. Okay, so phosphoenol pyruvate in this obviously with the phosphate group in the second carbon, with the removal of water, dying to give off this phosphate group, and that's what it does. And pyruvate kinase then, okay, pyruvate kinase removes the phosphate group to regenerate more ATP from ADP. Okay, and as we remove the phosphate group, we're producing pyruvate. Okay, and that's the final product of of glycolysis. Okay, again, this whole process, this reaction requires the involvement of not only magnesium, okay, but also manganese and potassium. Oops, it doesn't open there. Let's go back. Okay. Okay, so it requires manganese, magnesium, and potassium. These are all minerals. Okay, so again, relating it to diet. Okay, make sure when people advise people when they get advised to make sure that they have their full complement of minerals, usually by the consumption of fruit and veg. Okay, it's because these minerals are required as cofactors, as important molecules that facilitate these reactions. And again, it won't be just this reaction. There's thousands of other enzymes, hundreds of other enzymes, which require these minerals. Okay. Okay. So that's glycolysis. Let me just mention a couple of things about how it's regulated. Okay, I sort of alluded to the regulation already before. We know that hexokinase is a target for glucose 6-phosphate. Okay, if you get too much of this being produced, okay, you get the inhibition of hexokinase. Incidentally, too much of glucose 6-phosphate also is a stimulant for the production of glycogen as well. Okay, in the liver and in the muscle cells. Okay, so too much of this not only does it block hexokinase, but it also then is shuttled to produce glycogen. Okay, now phosphofructokinase and pyruvate kinase are also two further enzymes that are inhibited, that are targets for inhibition. Okay, but rather than the products inhibiting these enzymes, it is basically the concentration or the ratio between ATP and AMP. Okay, lots of ATP will feed back negatively to inhibit phosphofructokinase and pyruvate kinase. Okay, but if you have lots of AMP, okay, then it actually stimulates these enzymes to work faster. ATP inhibits and AMP stimulates. Okay. Okay. So now if we just take a step back and just look at the number of important molecules that are produced in these in glycolysis. Okay. So again, the energy investment phase, we put 2 molecules of ATP in. Okay. And then the subsequent half of the glycolysis pathway resulted in the production of 4 ATP. Okay, remember 2 molecules of ATP for every glyceraldehyde 3-phosphate, and because we get 2 3-glyceraldehyde phosphate molecules, 2 times 2 is 4. Okay, so we get a net So we get the production of 4 ATP molecules for every molecule of glucose. Okay, but because we put 2 in and we get 4 out, we're left with a net production of 2 ATP molecules. Okay. But probably what's more important in the context of cellular respiration as a whole is the production of this very important molecule. Okay, NAD to NADH. Okay, is the generation of NADH. So electrons are carried by NAD to form NADH. Okay, and it's these electron carrying molecules which are then subsequently used for other pathways further down the line. Okay. So I'm just going to skip through these questions. Okay. Okay, so a bit too fast. Right then, so after we get the production of pyruvate, okay, pyruvate cannot enter the citric acid cycle straight from there. It has to be converted to what we call acetyl CoA. Okay. So this is mediated by what we call a multi-enzyme complex, and it really performs 3 different reactions. Okay, we're not going to go into the reactions in any detail, but the first reaction here results in the production of carbon dioxide. So we're basically uh, starting to break this down already. Okay, so we've got a 3-carbon molecule of pyruvate. Okay, we're ending with a 2-carbon molecule attached to an acetyl, attached to a CoAsh molecule. Okay, a CoA molecule, and so as we break, as we remove one of the carbons, that gets converted to carbon dioxide. Okay, now as we break it down, as we oxidize it further, that energy is captured to reduce NAD to NADH. Okay, so we're getting the generation of another molecule of NADH. Okay, so if you remember, NADH is the energy carrying molecule that's used for subsequent processes in cellular respiration. Okay, uh, so we get the generation of that, and then the third bit of the reaction performed by this multi-enzyme complex is for the attachment of coenzyme A to this 2-carbon molecule. Okay, to produce the mature acetyl CoA molecule. Okay, and it's only in the form of acetyl CoA can this enter the citric acid cycle. Now, this name here might be familiar to you from a previous lecture, and if it is, okay, and if you can recall that we talked about acetyl CoA when we were talking about fats. Okay, when we how we generate fats is by the addition of acetyl CoA to glycerol. Okay, or to produce fatty acids rather, not glycerol. We attach acetyl CoA, okay, in the production of fatty acids. Okay, uh, so what this basically means is that instead of acetyl CoA being shuttled down into the citric acid cycle, it can be shuttled if there's already enough energy being produced. This acetyl CoA can then be used to generate fats. Okay, for storage. Okay, so acetyl CoA can be joined together to form fats, especially in fat cells and in the liver. Okay, and so so this is the transitional step. Okay, before we get to the citric acid cycle. Okay, I thought this is an important step to mention because without acetyl CoA, we don't get any input into the citric acid cycle. Okay, so pyruvate has to be converted to acetyl CoA before it can enter the citric acid cycle. Okay, now this is the citric acid cycle as I showed you before. Okay, now we're not going to go through this in any detail. Okay, oops, sorry. Okay, but let's just summarize this pathway. Okay, by saying that it the TCA cycle completes the breakdown of pyruvate, okay, to carbon dioxide. That's one of the reasons why we have the citric acid cycle. But the major reason why we have the citric acid cycle is the energy that's locked away in acetyl CoA, okay, is used to generate these really important molecules, NAD. So more of these important molecules, NADH and FADH. Okay, that's the principal reason why we have the citric acid cycle. This happens in the mitochondria. Okay, so here's the transition step. Okay, pyruvate being converted to acetyl CoA. Acetyl CoA then uh, combines with oxaloacetate. Okay, to form citrate. Okay, through this enzyme citrate synthase. So it's synthesizing citrate from oxaloacetate by combination of acetyl CoA. Okay, and then you get this molecule here, citrate or citric acid, if you like. Okay, uh, can you see over here? We've got three carboxylic acid groups. One here, second, third. Hence the reason why it's called the tricarboxylic acid cycle, because you start off with three carboxylic acids. Okay, and then that's further modified through the action of these different enzymes to perform to break it further down. Okay, further down until we get back to oxaloacetate. Okay. Now, as it's been oxidized, as it's been broken down by all these different enzymes, we start to generate carbon dioxide. Okay, so a molecule of carbon dioxide here, another one here. Okay, and also we had one in the transition step as well. Okay, so the actual generation of carbon dioxide happens at this step, at this transition step, and in the citric acid cycle. This is where the CO2 is generated. Okay, um, so I won't really say anything more other than that. One thing I will say is just to remember that this step here, succinate to fumarate, okay, and which results in the generation of FADH2, okay, another electron carrying molecule. This is also part of the oxidative phosphorylation pathway as well. Okay, so this step here is linked to the oxidative phosphorylation pathway, as you'll see in a few minutes. Okay, so just to reiterate, then we get the production of NADH. So most of the NADH is being produced at the citric acid cycle, but it's also the first cycle, or rather, uh, the first cycle if you consider the transition stage part of the cycle as where the CO2 is being produced. Okay, so when we talk about the cellular respiration pathway, glucose plus oxygen gives you ATP, H2O, and CO2. That CO2 comes from here. Okay, comes from the citric acid, comes from the transition stage. Okay, and the citric acid cycle. Okay. So just to summarize that, then, the TCA cycle has 8 steps, mediated by 8 different enzymes. Okay, the acetyl group of acetyl CoA joins the cycle by combining with oxaloacetate, forming citrate. Okay, and then the subsequent steps really are there to decompose the citrate back to oxaloacetate. Okay, and so therefore it's because it goes full cycle, it's called a cycle. Okay. Okay, so because the pyruvate, which initially enters this particular part of the cellular respiration pathway, okay, which gets converted to acetyl CoA, it's still considered an oxidizing part. Okay, so it's you get lots of oxidation here. Okay, so if things tend to be broken down, they're being oxidized. Okay, and in the process, you're producing these really important molecules, NADH and FADH. Okay, so for every turn of the cycle, you're producing 3 molecules of NADH and 1 molecule of FADH2, as well as a molecule of GTP. Now, GTP you can consider as ATP. They kind of do the same thing. Okay, so that's for every turn of the cycle. So in other words, for every in one molecule of pyruvate. So for every molecule of glucose, you're producing two molecules of pyruvate. So you will have two circles for every molecule of glucose. So you can double this then. Okay, so you're going to produce 6 molecules of NADH, 2 of FADH2, and 2 of GTP or ATP. Okay. So citric acid cycle really important in generating these molecules, these electron carriers. Okay. Okay. Now, in terms of regulation, we know how glycolysis is regulated. Okay. Similarly, the TCA cycle can also be regulated as well. Of course, when you have lots of ADP and lots of NAD, this basically means it's a sign that there's not enough energy, not enough ATP being produced. So lots of ADP and NAD will increase this pathway. Okay. And ADP and NAD will activate these particular enzymes, isocitric dehydrogenase and alpha ketoglutarate dehydrogenase. Okay. Um, and this pathway will be reduced in terms of its speed. Okay. If there are high levels of ATP and NADH. If you've got lots of these molecules, that means there's lots of energy floating around. There's lots of ATP knocking about, so therefore we don't need to produce much more. So that's the signal. That's the way that's the way in which this particular pathway can be slowed down. Okay, and ATP and NADH are targets for isocitric dehydrogenase. Okay, uh, and as soon as these two molecules bind, this enzyme is inhibited. Okay. Um, it also decreases when there are high levels of NADH, as we know from here. Okay, or succinyl CoA. Okay, succinyl CoA, which is one of the products. Okay, and both of these, so NADH, not only does it inhibit isocitric dehydrogenase, but it also inhibits alpha ketoglutarate dehydrogenase. Okay, as well as succinyl CoA. Okay, so when you have lots of these molecules floating around, lots of succinyl CoA, that means there's lots of the pathway is running quite quick. That means lots of ATP is being generated. Okay, and when you start to get build up of succinyl CoA, lots of NADH, that means there's just too much energy in the system, so it needs to slow down. Okay, hence the reason why these are targets for inhibition. Okay, so okay, just to mention those important molecules again. Okay, so you get one molecule of NADH here, another molecule of NADH there, a molecule of ATP or GTP here, and one molecule of FADH2 there, and another molecule of NADH. Okay, so uh, this is for every one turn of the cycle. So because we have two, because there's two molecules of acetyl CoA coming in, so we can double this. Okay. So when we start to take stock then, okay, of the TCA cycle, uh, it you can see that we produce two molecules of ATP or GTP, 6 molecules of NADH, and 2 molecules of FADH2. So remember, this is for every initial glucose molecule we put into the system. For every initial glucose molecule, you're producing 2 molecules of pyruvate, and therefore 2 molecules of acetyl CoA. And therefore, when you combine the two, this is what you get. Okay, 2 of ATP or GTP, 6 of NADH, and 2 of FADH2. Okay, and so just to mention that point again about NADH and FADH2 being the very important electron carrying molecules, okay, which are really important for the subsequent step of oxidative phosphorylation. Okay, it's these molecules which drive the next pathway. Okay, so it's imperative that we produce. If we want to produce lots of ATP, then it's imperative we produce lots of these molecules here. Okay, so put it in a table form. As far as glycolysis was concerned, we the energy investment phase we put 2 in, and then the energy payoff phase resulted in 4 molecules of ATP. Okay, um, TCA cycle, we didn't put any energy in, but we still got 2 molecules of ATP out. Okay, so 4 minus 2 is 2 plus the 2 there, so the net gain is 4. Okay, as far as the electron carrying molecules are concerned, okay, the NADH, we get 2 in glycolysis, 6 in the TCA cycle, and 2 in the pyruvate oxidation pathway. Okay, which gives us 10. And then FADH2, we get 2 only in the TCA cycle. So in terms of electron carrying capability, we're getting 12 electron carriers. Okay, 12 electron carriers when we take stock after these the 2 main cycles so far, glycolysis and TCA cycle, and uh, the transition step, the pyruvate oxidation to acetyl CoA. Okay, right then. Okay, so this then brings us to the last step, the last stage, the last pathway, if you like. Okay, the oxidative phosphorylation pathway. Okay. Now, this is the reason why we need oxygen. Okay, why we breathe in oxygen. If you don't have enough oxygen in the system, this pathway gets blocked. It gets backlogged. Okay, and we can't produce ATP, and the whole system then comes to a halt. And it doesn't quite come to a halt, but it definitely slows down. Okay, so oxygen is really important here because, as I've said many times before, it is the molecule which is the acceptor of the electrons at the end. Okay, so oxygen accepts the final electrons from NADH and FADH2. Okay. Most of the ATP that we get produced in cellular respiration happens here. Okay. What is oxidative phosphorylation? Okay. So it happens in the mitochondria. Again, we know this now. We should know this by now. It is oxidative because the process is taking place in the presence of oxygen. Okay. And phosphorylation, so oxidative phosphorylation. Phosphorylation because the final step in the process, okay, is mediated by a kinase. Okay. Which what does a kinase do? It phosphorylates. Okay. So it's phosphorylating ADP to produce ATP. Okay, so it's a phosphorylation process. Okay. Oxidative phosphorylation involves electron transport, as you'll see in a minute. Okay, and chemiosmosis. Okay, and it requires an adequate supply of oxygen. Okay, any of these things go missing. Okay, we don't get oxidative phosphorylation. Okay, so if something happens to the electron transport chain, and we can't generate, you know, we can't perform chemiosmosis. Okay, or we don't have enough oxygen being brought in. Okay, then we stop or we slow down oxidative phosphorylation. Okay, now the electron transport chain is in the inner membrane, the cristae. Okay, so here we are, the cristae. So these inner folds. This is where it takes place. This oxidative, well, most of the oxidative phosphorylation. Okay, in the mitochondria. Uh, the electron transport chain is basically a collection of protein complexes. Okay, and we have complex 1, 2, 3, and 4. We have another protein at the end, the kinase, but that's the production of ATP solely. Okay, uh, as these complexes basically are proteins, are a collection of proteins which, uh, a bit like a relay system, they relay the electrons. Okay, and as they relay the electrons, they harvest some of the energy. Um, and use that energy to pump to pump hydrogens or hydrogen ions out into the intermembrane space. Okay, so the carriers alternate reduced and oxidized state. So again, what is oxidation and reduction? It's the loss and the gain of electrons. So one complex is gaining electrons, and then it donates electrons to another complex, and so you get this constant redox pathway. Okay. And as they go from one complex to the next, okay, electrons drop in free energy. Okay, so as they go down the chain there, and once they get to the end, to complex 4, okay, that's when they really have no more energy. Well, the amount of energy left in those electrons is quite small. They're still obviously negatively charged. Oxygen accepts those final electrons. Oxygen becomes really electronegatively charged then, and therefore it reacts with hydrogen to form water. Okay. Okay, so I've mentioned this word before, but I haven't quite described it properly yet. So chemiosmosis is the movement of ions, and particularly hydrogen ions in this case, okay, across a semi-permeable membrane. So it's a bit like osmosis, okay, the movement of water, but rather than water, we're moving ions across. Okay, and because we move them into the intermembrane space, we're sort of concentrating hydrogen ions in one part of the membrane, on one part of the mitochondria. Okay, and there we create this electrochemical gradient, high concentration in one place, low concentration in the other, and so we produce this electrochemical gradient. Okay, and it's this gradient that we generate, and then through the diffusion of or reduction in this electrochemical gradient by diffusion generates the kinetic energy to produce to regenerate ATP. Okay. Um, so this bit I'm gonna I'll just say it now, but I'll explain it a bit more. Okay, as the electrons are passed from one complex to the next, okay, the high energy electrons, or the free energy, is quite high in these high energy electrons, and it's these that give the energy for those complexes to pump hydrogen ions across the mitochondrial inner membrane into the intermembrane space. Okay, so like I said before, this is what generates this electrochemical gradient. Okay, or you could call it even a potential difference, or a bit like a voltage. Okay, and as the hydrogen ions return back across the inner membrane, the energy that's provided by this movement, okay, drives the ATP synthase to phosphorylate ADP to generate ATP. Okay, so okay. So again, this is a bit of repetition. Let me just go through it. Okay, so these high energy electrons generated, carry obviously in glucose. Glucose is broken down. Okay, it's oxidized in the glycolysis pathway and in the citric acid cycle pathway. And not only are you generating a few molecules of ATP, but more importantly, you're generating these molecules, these electron carrying molecules, NADH and FADH2. Okay, and these are the molecules responsible for carrying these high energy electrons. Okay, those electrons at the end of the whole pathway, of the whole oxidative phosphorylation pathway, get picked up by oxygen. Okay, so these electrons get picked up by oxygen, react with hydrogen ion to form water. Okay, um, and it's this energy that's released from these redox reactions. Okay, passing electrons from one complex to the next, that results in those complexes being able to pump hydrogen from the mitochondrial matrix into the intermembrane space. Okay. And just to repeat again, it's these hydrogen ions that diffuse back across the inner membrane, which give the ATP synthase the energy to synthesize ATP from ADP. Okay, so there's quite a bit of repetition there, but just to hammer down the point. Okay, so this is the electron transport chain. Okay, and I'll illustrate what chemiosmosis is at the same time as well. Okay, so complex 1, complex 2, complex 3, and complex 4. Okay, and this is ATP synthase. Okay, so here we have it. Here we have the NADH. Okay, NADH, and it's electron. This is carrying high energy electrons. It interacts with complex 1, okay, through this flavin nucleotide. Okay, this picks up the high energy electrons and then shuttles them across. Okay, shuttles them across to other components in the complex. Okay, and you can see over here, that these electrons are just being shuttled from one place to the next. Okay, so the energy that's carried in these high energy electrons, okay, is used to pump uh, protons. So when I say hydrogen ions, I'm talking about protons. Okay, what's a proton? It's just one proton, no electrons. What's a hydrogen ion? It's just one proton, no electrons. It's the same thing. Okay, so for every molecule of NADH that gets oxidized, okay, this gets reduced. For every molecule of NADH, we're able to pump 4 hydrogen ions, okay, into the intermembrane space. Okay, NADH then becomes NAD. NAD then is then free to go back to glycolysis, okay, to generate for it to be used in glycolysis. Okay, not only in glycolysis but also the citric acid cycle as well as the pyruvate dehydrogenase steps. Okay. Anyway, uh, these electrons then are shuttled across and picked up by coenzyme Q, which then shuttle them across further into complex 3. Okay, but we've missed out complex 2 here. Okay, now let me explain why that was the case. Okay, because what I did mention in the citric acid cycle, that there was one stage I wanted you to bear in, just to remember. It was the formation of fumarate from succinate. Okay, succinate from fumarate was part of the citric acid cycle, and as a consequence of that, you also produced FADH2 as well. Okay, so again, high energy electrons being carried by FAD. Okay, so complex 2 isn't ignored. It's just a bit staggered. Okay, the pathway is a bit staggered. So you get electrons being brought in from NADH, and then through the citric acid cycle, getting electrons being carried by FADH2, which are then transported to the coenzyme Q, okay, which are then transported to um, the complex 3, okay, and then again high energy electrons, lots of energy there, and it's then used to generate some energy to pump for complex 3 to pump another 4 ions of hydrogen. Okay, so what you'll notice is that there was high energy electrons in NADH and high energy electrons in FADH. Okay, so we've used up large amounts of the energy already to pump 4 hydrogens from complex 1 and 4 hydrogens from complex 3. By the time we reach this stage here, we don't really have that much energy left in these high energy electrons. Okay, and that's partially the reason why we're only able to generate enough energy to pump now 2 ions of hydrogen. Okay, before we had enough energy to pump 4 here, 4 there, but now we only have enough to pump 2 ions of hydrogen. Okay, so okay, so before I talk about the next bit, okay, so that's a bit of repetition. I just talked about this, so I'll give that a skip. Okay. One thing about this protein here, cytochrome C, which you'll find in all cells. Okay, it's a mitochondrial protein. Okay, but it's one of those. It's a protein which it's an enzyme, cytochrome C oxidase, ASE at the end, so it's an enzyme. Okay, and it catalyzes the oxidation of the cytochrome C. Okay, so cytochrome C oxidase. Okay, but it's also involved with shuttling electrons from complex 3 to complex 4. Okay. Right then. One thing I wanted to mention here was that when we talk about carbon monoxide being a toxic gas, and we know you probably heard stories about cyanide. Okay. Um, Probably seen it in movies when people take cyanide and people start frothing from their mouth. Okay. Cyanide is very fast acting. Okay, so people were poisoned with cyanide. It's very fast acting. Yeah, the cyanide gets into every cell systemically, and basically you stop producing ATP. Okay, carbon monoxide is a silent killer. It's a slow and silent killer. Okay, if your gas boiler isn't working properly, and so the gas isn't being combusted to CO2 and gets partially combusted to carbon monoxide. Okay, carbon monoxide is something. It's a gas which you obviously can't smell, and so you breathe it in, and just slowly, 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 it takes over all your cells. And the way it works is that both of these act on cytochrome C oxidase. Okay, so basically you block this final step into complex 4. Okay, and so there, because you stop that, you don't get the subsequent generation of hydrogen into the intermembrane space. Okay, you don't get these electrons being donated to oxygen to form water. Okay, and therefore it's lethal. Okay. Okay, so after the pumping of these hydrogen ions into the intermembrane space, okay, this is what happens as a result of that. Okay, so you get a lot of hydrogens being pumped into the intermembrane space, so you generate a gradient. Okay, lots of hydrogens, lots of protons on one side, and not so much on the other side. So we've got lots of positive charge on one side and a relatively negative charge on the inside. Okay, this should be charge. Okay, and so because you've got this electrochemical uh, charge difference, okay, and obviously diffusion forces all these hydrogens then to find a way to get into the matrix. Okay, luckily we have this complex here, this protein complex called the ATP synthase, and hydrogen ions are then are allowed to diffuse through 3 at a time. Okay, to generate the kinetic. This is a bit like a rotor system. Okay, it turns round and round. Okay, a bit like a dynamo. Okay, and as it goes round and round, it's able to provide the energy needed to regenerate ADP to ATP. Okay, and this is the ATP synthase. Okay, and this is where you get the vast majority of ATP being produced. Okay, through this last step. So if you were to inhibit this step here through cyanide and carbon monoxide, you're not going to get the ATP production here. Okay. So, and through the oxidative phosphorylation pathway, you're getting about 26 molecules of ATP. Okay, just through that. Okay. So when we put it all together, then in terms of the ATP, this is how much we're generating, about 32 molecules of ATP in terms of the net gain. Okay. Obviously, we put 2 in, but we get 4 out here. Okay. So 26, uh, 28, 30, 32. Now, this is kind of an experimental value. Okay. We can go up to even 36 molecules of ATP. So it just depends on the cell type and the conditions. Okay, if they're perfect conditions in the perfect cell, in the cell like a muscle cell or which has lots of mitochondria in, you can actually generate more ATP for every molecule of glucose. Okay, but on average, if you take all the cells and take an average, we tend to produce around 30 to 32 molecules of ATP for every molecule of glucose. Okay. And just bear in mind that most of the glucose is being generated here at the oxidative phosphorylation pathway. Okay, the TCS cycle, the pyruvate oxidation, and the glycolysis is really there to generate the important NADH and FADH2 electron carrying molecules. Okay. So just to finish off then. So I'm just going to leave you with this last slide now. Okay, just to point out that electrons transport is coupled with ATP synthesis. Okay, so ATP synthesis can only happen when electron transport chain is working properly. Okay, without that, ATP synthesis doesn't occur. Also, if you think about it, ATP cannot be synthesized by oxidative phosphorylation unless there is energy from electron transport. Now, what that means basically is that the electron transport chain carries and donates these high energy electrons from one complex to the next. Okay, so it's the energy carried in these high energy electrons that mediate, that facilitate rather, okay, the oxidative phosphorylation pathway. Okay, so you need the energy from the high energy electrons in the electron transport in order for ATP synthase to actually produce ATP. Okay. Important substrates, obviously we know now that NADH and FADH2, oxygen is an important molecule because because it's the final acceptor of the low energy electrons. ADP, we need plenty of that floating around in order to generate that, in order to phosphorylate that to produce ATP. Okay, and how the whole thing is regulated is really by the ratio of ATP and ADP in the whole mix, in the whole cell. Okay, lots of ATP, that means we don't need so much, so we slow down this the whole cellular respiration pathway. Lots of ADP knocking about, then it's a sign that we need more ATP, so that ADP tends to speed things up. Okay. Right then, it says over here high ratio inhibits oxidative phosphorylation. Okay, because ATP. So if you have lots of ATP, we don't need to produce ATP. And how it works is that we can have the complex 4. So if I go back a couple of slides, okay, complex 4 here, ATP can bind to this and change its structure. So when we say allosterically, okay, allosterically binds, changes structure, and inhibit this complex. Okay, so that's the regulation of oxidative phosphorylation. Okay, so these molecules are there to regulate this process. Right then, that's it. So there's all these questions. Again, you can go through them in your own time. So let me just go through that and finish off with the learning outcomes again. Okay, here we are, the learning outcomes. So again, we've talked about cellular respiration. Okay, we've looked at glycolysis in some detail. Okay, we've looked at the production of ATP as well as other important molecules throughout the different stages of cellular respiration, and we've also looked at how these different stages can be regulated. Okay, and of course now you should know by now that this whole process is facilitated by redox processes. It's all redox reaction. Mostly it's all redox reactions, just moving electrons from one place to the next. So oxidizing one, reducing the other. Okay, so redox reactions. So there you go. That is the end of it all. Okay, that's 14 lectures we've had. Okay, we started off with redox in the first. We started off talking about oxidation and reduction reactions. We talked about electrons in the beginning, and that's where we kind of finished off as well. So, okay, so we've come full circle. Okay, talking about electrons, redox reactions in the context of cellular respiration. Wonderful. Right then, so that's it. Again, if there's any questions, if there's anything you don't quite understand, something maybe you think I've missed out, please send me an email, give me a shout, somehow contact me, and I'll answer the questions. Okay. Okay then. Well, I then now bid you all the best for the upcoming exam, the final test in January. And yep, until I hope everyone stays safe and enjoy your Christmas and have a good break. Okay. All the best. Bye bye.